Hey, listeners of the Bio Report. Before we get started this week, I wanted to tell you about the digital library from Deep Dive. How much time does your team spend looking for research papers? Google, PubMed, social media. There's got to be a better way. You can now search a reference database of 100 million scientific papers and read the full text of 20 million articles, annotate them, and share with colleagues. It's the smarter way to do research. Here's the best part. If you're like me and been frustrated by not being able to access articles you find because they're behind a paywall, I've got good news. With Deep Dive, you get one-stop affordable research. If you're a listener of the Bio Report, you can try the enterprise version of the service for free for 1 month. Go to deepdive.com/podcast and enter the code BioReport. That's Deep Dive, DeepDive.com/podcast. And the code is BioReport, one word, all caps. I'm Daniel Levine, and this is the Bio Report. While the class of immunotherapies known as checkpoint inhibitors has brought a promising new approach to treating cancer, the development of resistance to these therapies limits the number of patients they benefit. Portage Biotech is building a set of platform technologies and a pipeline of immuno-oncology therapeutic candidates to address this problem. We spoke to Ian Walters, CEO of Portage, about the problem of resistance, the company's pipeline of candidates, and its portfolio-based business model. Ian, thanks for joining us.
Hi, I'm very pleased to be here today and to share a little bit more information about my company, Portage Biotech.
We're going to talk about Portage, its pipeline of immuno-oncology therapies, and its business model. Prior to Portage, you were at BMS and involved in the development of some of the first immunotherapies known as checkpoint inhibitors. Perhaps we can begin with this general approach. What happens in cancer and how do these therapies work?
Sure. So for many years, you're familiar, when we were treating cancer, we would give people toxic chemicals. They would wreak havoc throughout the body with the hopes that you'd get small amounts to your cancer and that could slow the growth. And some people often complain that the treatment is worse than the disease. You know, at the early days at BMS, we had made the shift to try to boost an immune system to fight cancer. And the reason that I don't have cancer today, even though my body develops cancer cells every day, is because my immune system finds those cells, it sees them, and then it clears them. So the idea being, what if we can get everyone's immune system to find the cancer cells and kill them? So we started using some, I would say, rather broad and non-specific approaches to boost the immune system. And when the immune system fires at the cancer, if it can recognize a cancer cell over a healthy cell, it can clear that cell, and it can continue to do that for typically the remainder of people's lives. And it really was a shift in how we approach cancer. And we're, for the first time, we're really seeing patients with very long response after stopping treatment. One of the drugs that we were developing at BMS was ipilimumab. That's a checkpoint drug. And we saw the patients that were in response after 1 year were still in response after 10 years and are effectively cured. And that really created a paradigm shift in the treatment of cancer. And many, many companies now are developing treatments in this area.
Well, these therapies have been effective for some patients. They don't benefit a large portion of cancer patients with a given condition. Why is that?
Yeah, so there's a variety of reasons. So let's start with the two tumor types that are the most sensitive to immunotherapy: non-small cell lung cancer and skin cancers. It's not surprising that these are responsive to immune treatments because they're also two of the most highly mutated tumor types. So when we're outside, we're exposed to radiation that causes mutations in our skin cells. And skin cancer typically has a lot of mutations. So it's very easy for the immune system to recognize what's a cancer cell and what's a healthy cell. Same thing with lung cancers. You're breathing in toxins and things from the environment. Lung cancers tend to be one of the also highly mutated tumor types. It's very easy for the immune system to recognize. When you look at other tumor types, let's say pancreatic tumors, those are very low mutated. And it's not that easy for the immune system to recognize what's cancer and what's a healthy pancreatic cell. So they don't typically respond. So that's one of the key features that's needed for these treatments to work, right? We need to be able to understand, and the immune system needs to be able to recognize what's a healthy cell and what's a cancer cell. So that's one of the key reasons a lot of tumor types don't respond, even in skin cancers or lung cancers. If they have a low level of mutations, and this is something that we can test in patients, these typically don't respond. So that's some of the reason, the ways that we're looking to augment the existing drugs is by helping the immune system recognize what's a healthy cell and what's a cancer cell. There's also the notion that these drugs really relieve something what we call the break signal. So a cancer cell can express a certain protein on the surface of its cell that tells the immune system, hey, I'm a good cell, you can leave me alone, and it prevents the immune system from clearing it. So these treatments can remove those signals, and which enables the immune system to attack the cancer. Now, not all tumors express those markers, and that's another area of focus of how we increase the expression of these markers on the surface of the cancer cells to allow the immune system to recognize it and clear it. So there's a variety of different biologic mechanisms that contribute to response, and why typically with the existing drugs we're seeing as a single agent, somewhere between 10 and 30% in different tumor types respond. Sometimes the addition of one or more agents together, you can get more patients to respond. On the converse, there's 70% of patients that are not responding. And that's the big area of focus of what we're doing at Portage.
How well understood is resistance with regard to immunotherapies?
Yeah, so like I mentioned previously, in the early days, we were really excited about these treatments, and we were hopeful that we would figure out ways that everybody could respond. Unfortunately, there are a variety of reasons that these either develop resistance over time or never respond in the first place to these drugs. And this is something that we started learning about 10 years ago when we first started testing these drugs. And me, as well as members of my team, were part of that group at BMS in the early days doing these testings. So we took a lot of these learnings with us, and has really been kind of our marching orders to identify new products that could overcome these resistance, whether it's the mechanism we talked about, which is immune recognition, how to make sure the immune system can recognize the cancer, or how you can take an existing tumor that doesn't have any immune response already in the tumor, and to try to make that tumor hot, or what they call immunologically hot, which means we get immune cells to get into the tumor. And there's also the microenvironment around the tumor. So again, there's all kinds of ways that tumors avoid killing by these methods. One of them is they create sort of a hostile environment for the immune system. They have a variety of different negative cells. We call them like the army that are protecting the tumor. And we are also looking at strategies how to take out that army so that the immune cells can get in and do their job and clear the tumor.
Portage has a number of platform technologies. I wanted to focus on your invariant natural killer T cell agonist. These provide a way to activate both the adaptive and innate immune systems in the fight against cancer. Can you explain what this is and how it works?
Sure. So we're going to have to get into some hardcore immunology in order to explain this. So, the immune system has a variety of different ways that it protects the body. So it has an innate immune pathway, and that's sort of like the first line of defense. Those are the immune cells that sit in tissues and that guard your body against various different abnormalities, whether that's infections, cancers, and other things. And, for a long time, people didn't even care too much about that arm of the immune system because we didn't have really good tools to kind of stimulate it. The invariant natural killer T cell, I'm going to call it iNKT for short, is a type of cell that is able to stimulate that arm of the immune system. And just to give you an example of how that works in real life, the way we understand these things typically is we take a mouse and then we remove the cell and we see what happens. So if you take out in an animal the ability to make these type of cells and you give it a cancer exposure, those animals will develop cancer, while an intact animal that has these cells will be able to clear the cancer challenge. So that gives you a sense. These cells sit in tissue. They are recognizing cancer cells as they enter the animal, and they're able to clear them. And if you don't have those cells, you will not succeed in that, and the animal will develop a tumor. So that's immune surveillance. That's the innate part of the immune system. So that's only one bit on the mechanism of action. The second bit is the adaptive immune system. And that's, again, and in today's day and age, a lot of people are learning about the immune system with respect to COVID. And you probably have heard people talk about T cell responses to COVID. Well, the adaptive immune system is designed to ramp up when there is a specific abnormality, whether that be cancer or a virus. And this particular cell in our products are able to ramp up that adaptive response and create a broad immune response that includes T cells and B cells and antibodies, all to fight the cancer as well. So you get really two different plans of attack to try to improve the situation for cancer patients.
You have two versions of your iNK T cell agonist in development. This is port 2 and port 3. My understanding is they differ in the way they're packaged and delivered. Can you explain the difference between these two experimental therapies?
Sure. So in order to improve these particular drugs based on their chemistry, how they circulate the body, where they go, we have decided that we want to package them. And there's two different ways that we've come up with to package and deliver them that give them the ideal properties for a therapeutic. So port 2 is an iNKT agonist in a nanoparticle. In a liposome. Very similar to how they deliver a COVID vaccine, right? A liposome. And what that does is that helps direct these things to the tumor over the healthy tissue, and it gives them the ability to circulate longer without being naturally broken down by the body. So you can give them less frequently. So that is port 2. Now, port 3 is part of a collaboration we have with a few immunologists in Europe who did some work about a nanoparticle formulation. And that's another kind of encasement around the drug. And what they studied there, and this enabled us to get a grant from the EU to study this particular asset, was that when you put our iNKT agonist with a antigen, which is a part of a tumor, like, to use the analogy in COVID, they talk about the COVID vaccines have a spike protein, right? Here we're using a part of the tumor plus our agonist in a nanoparticle. And when you put those two things together, they're 5 times more potent than giving the two products individually. right? So we get a real interesting synergy by combining those two aspects in one product. Also, locates to the tumor and helps bring these drugs exactly to where they need to be in order to start battling your cancer. So we've come up with these two kind of creative ways, and we've developed some IP. We've gotten support from the EU and grants and things to develop these two different formulations in order to see if we can find better ways to treating different cancers.
And where are you in development on these, and what's known about their safety and efficacy to date?
Mm-hmm. Well, we announced earlier this year in April that we started our trial with Port 3 in the EU. And it's early days. We haven't really reported any of the human data yet. And we're just about to start our trial with Port 2 also in the UK. So I can't give you a whole lot of information about safety and efficacy in humans. That being said, we have seen really dramatic efficacy of these agents in the typical mouse models. And one of the things that we're really good at, having worked in this area, since the very early days, is knowing which animal models are most likely going to translate to what you might see in humans. So we've seen incredible activity in the mouse models of these two. And there's been other kind of agents that are attempting to do a similar thing with this cell population, done by other companies, which have shown, remarkable safety in humans. So I think it is our expectation, obviously we need to do the studies and collect the data, that there's a good probability that these will be well tolerated, and we'll have to see if our novel way of delivering them and the formulations and the chemistry will lead to, a really interesting efficacy signal in humans as we see in the mouse models. So you're going to have to stay tuned, and come back to me in 6 months to 2 years when we have more data with these assets to get the full picture.
These have the potential to be broad-acting therapies. How do you go about selecting and prioritizing the indications you're going to pursue?
Yeah, so this is a little bit of an art in drug development, right? Because there's a lot of features and factors that you have to consider to try to understand how a drug works and where it might work and what's the best opportunity from a medical, scientific, and business perspective to pursue. Now, with these therapies, some of the data that we have demonstrated in animals, which really helped us decide how to do this in human testing, was their ability to work in conjunction with those same drugs that I developed when I was at BMS. So there's a class of immunotherapy. It's the widest used product right now in oncology, called an anti-PD-1 inhibitor. That's a checkpoint drug. That's one of the drugs that me and my colleagues helped develop when we were at BMS. And that drug is doing extremely well. That class of drugs has been approved for something like 18 different tumor types and is currently selling over $20 billion a year when you add up the sales of the different companies developing these drugs. And just about every major pharma company has one of them approved. So what we've shown is we can, in animals that don't respond to that drug, we work as a single agent, right? So we could certainly go in conditions where patients have failed those particular drugs, and that's part of our clinical strategy. And we're doing that in the context of lung cancer and skin cancer. Again, some of the areas that we know are typically responsive to these drugs. And then we also see when you give the combination of our iNKT plus these PD-1 antibodies that we can restore sensitivity to those. And there's really good scientific data on why that's the case, how we change the surface of the cancer cell, how we impact the immune cells. So that's another area of our clinical trial where we're looking at our drug alone versus these PD-1 drugs alone versus the combination of the two to see if we could enhance the activity. So, there was pretty good scientific evidence on where to go and what indications based on the use of these PD-1 agents in different tumor types. And that's really what we started with, which was the basis of the science and the medicine and what we know about the current treatment, and then extrapolating where we think our drug could best fit in, where the competition is, and where we could be, leaders in the area.
Portage is not a traditional discovery and development company. It's using a portfolio approach to acquire assets and develop them to license or sell, with the goal of maximizing potential returns while eliminating a lot of overhead biotech companies face. What does Portage look for in the assets it acquires?
Yeah, so we've gone out with specific scientific hypotheses that we want to solve. And that work really started back when we were at BMS and when we were looking, not only did I work on the drug development side and overseeing the clinical trials at BMS, I also worked on the business side as well, and was looking externally to BMS at assets to bring in. So I started looking in this space, many, many years ago, and trying to understand the scientific areas that were most appealing. And some of those tend to be what we call in the white space, right? So in areas that no one's currently looking at, right? Because we don't want to be the 10th person looking at one particular biology. We want to be kind of the pioneers and the first people who have access to a specific technology if we think it has value in the treatment of cancer patients. So we really looked quite broadly for assets in this space that could meet those criteria, that could be first in class or best in class, that represent some of the areas that are underdeveloped, where we could be in our own lane as far as the development and so on. And, part of that really involves a deep, deep understanding of the entire landscape, of what all the companies, big and small, are doing, and how we can find niche areas where we can be successful. So yes, we've been very targeted in how we've brought in products and platforms that address, key unmet needs in this space. And we've also been kind of intelligent in the way we develop them. So I think, it's pretty typical for new companies being started in the biotech space for them to raise a lot of capital, to hire a ton of scientists, to open up a big office in Cambridge or San Francisco, San Diego, raise more money, and build this tremendous organization with the hopes of trying to get products into the clinic that can help cancer patients. We've kind of stripped that down to the bare essentials, of what's really needed based on our, over 20-year experience in this space, where we don't need to hire a ton of scientists because we partner with lead scientists around the world. We keep our overhead down. We know the critical experiments. We've been on the business committees of major pharma companies who could acquire these products, and we really know what good looks like. We've said no to hundreds of companies in this space because they didn't have the right experiments or they didn't have the right information. So we really know how to streamline our efforts to try to do the experiments that are most valuable. So our investors really who put up the money don't spend a lot of money in infrastructure and benefits and things like that. The money goes directly to value creation R&D. And that's been our approach. And we've seen really good reception out there with either academic groups or small biotechs that really don't have this experience in-house and are very eager to partner with us to try to help them develop their products and get them to these value creation points as quickly as possible.
So I'm wondering if you can expand on that a bit. Tell me how you leverage your resources and minimize your overhead through the structure you've created.
Yeah, so, we're mostly a virtual organization. And we're able to split people resources across a lot of different projects simultaneously. So just to give you an example, we have one CFO who provides the financial oversight to a lot of different development programs that we oversee. So you don't need to have 6 CFOs. We don't necessarily, in most cases, have our own laboratories. That's a lot of infrastructure cost, right? We leverage exclusive relationships with different academic groups, with contract research groups to conduct our experiments so we can flex up or down as needed without necessarily needing to take on fixed costs. So we do things like this in order to, like I said, streamline the effect. And I think the real streamlining comes from knowing what experiments to do when, right? I think a lot of scientists in this area get sort of paralyzed in this paradox of, well, we need more data in order to move forward. And they keep doing more and more experiments, and each experiment yields 10 new questions and 10 new experiments. And you can get into this loop where you're constantly spending money doing more experiments and not necessarily adding significant value to your program. And the value really is getting it into the right human trial and knowing what experiments are needed to do that. And at some point, you have to say enough is enough. We're putting it into the clinical trials, and we're going to, move this into the most relevant species, which is human testing. So really having that insight and those experience creates probably the biggest, I would say, cost savings going forward, and that we're not doing glorified science experiments that don't really change your path forward.
And how far do you typically take an asset in development?
Yeah, so look, from a business perspective, and we didn't go over my background. It's a little bit unusual. I'm trained both in the medicine as a doctor and the science as a scientist, and I also got my business degree. So I kind of bridge all 3 disciplines. And from a business perspective, you have to be prepared as a group to take it all the way. Because as soon as you are in a situation where you don't have a path forward, that's when other companies will step in and take advantage. So I will give you an example, and this I think will kind of illustrate my point. So, ideally, we'd like to take a drug into human testing, show the safety and the efficacy, and then partner a license with a big pharma company, right? And that's kind of our standard paradigm for doing this. But if a pharma company doesn't believe that you're going to take something forward, you're not going to be able to negotiate the best deal. So you always have to be prepared to take things forward to registration, to commercial launch, and so on. So one of the first companies that we helped start and got going was a company called Biohaven. They're a Connecticut-based company. They are now at an $8 billion market cap, and they have one product approved that they're selling. And even as a small company, they're competing against big pharma and doing better in the sales and marketing of their agent. But that was a company where you would think, all right, well, once you have an approvable drug, you're going to partner with a big pharma company because a small biotech is not likely going to go out and sell a product. Their product is approved for migraines in a medical field like that where you need a big sales force and it's hard to commercialize. And in fact, they did it, and they did it well. And their valuation has appreciated dramatically as a result of that, and not necessarily relying on a partner and another company to come in and to do that for them. So our strategy as far as how far we develop is we're prepared to go all the way, even commercially market and sell a drug, but we're going to entertain offers from pharma partners all on the way. One of our value propositions as a company is that our group has been around long enough that we know everybody that's working in this space. There's really a set of target companies that are most likely the ones to be interested in our products, and we have ongoing relationships with them. We meet with them on a regular basis to kind of share our progress, to get ideas of interest, to understand what data they need to see to want to have these drugs in their portfolio. So we have a very clear sense of that. And then it's just a matter of executing and generating the data. So that's really our approach, and that we hope that that'll attract early interest from pharma companies. In fact, one of our companies has collaboration agreements with the two biggest players in this space, Bristol Myers and Merck, and we have joint development teams with them so that they get early access to see our data.
How do you think you might think differently about creating value under your business model than you would if you were a more traditional biotech?
Yeah, I mean, I think, most traditional biotechs are single asset, single platform companies, right? So they sort of live and breathe by a single program. For an investor, that's could be good or bad. If you have a big portfolio, that's not so bad. If a particular product or company works and you get big upside, or it doesn't work and you lose everything, right? Our portfolio approach is a little bit different than that. Since we're not beholden to just a single product, we have a whole pipeline of programs. We are constantly doing the business exercise of trying to understand where is the best use of our resources and how do we prioritize the programs and projects that are going to give us the biggest return on investment. And overall, that kind of smooths out the bumps in the stock. It sort of gives you multiple choices and multiple chances to succeed. In fact, in the next 18 to 24 months, I think we have 15 different trials that are going to be yielding data in that time frame. Any one of those could be a huge success and reproduce the success that we had with the company Biohaven. So that's really the main difference with our model, and that gives our investors really a significant chance for upside, regardless of one particular technology is succeeding or not.
Ian Walters, CEO of Portage Biotech. Ian, thanks so much for your time today.
It's my pleasure, and I'm happy to kind of circle back with you when we have more data to share about these exciting new agents.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.